Okay, so now in part three, we're going to talk about eukaryotic chromosomes because with bacterial chromosomes, they have really important compaction techniques, but you're compacting one individual chromosome. If we're talking about eukaryotic chromosomes, now we've got multiple chromosomes, and then those chromosomes are also linear, whereas with the prokaryotic chromosome, remember that they're circular. And so, if we talk about the set of chromosomes that would be in a human cell, for example, they would be over 1 meter long. So that means you take all 46 chromosomes, you stretch them out, and then you put each of those linear chromosomes next to each other to make one really long string. Again, it would be over 1 meter long, and that's inside of one of your cells. But you've got multiple cells inside of your body, obviously. So that tells you something about how tightly they must be compacted. But we need a really efficient way to both compact the information, make it smaller, but then we also need to be able to unfold that compaction so that we can actually reference those base pair sequences to build enzymes, right? And so remember that the DNA is never going to be found naked inside of the cell as long as the cell is alive. And those proteins that help compact, that help fold it, are in a really important part of what makes up the chromatin. Remember, chromatin is DNA plus proteins. So the specific proteins that we're going to use to wind up and wrap DNA inside of eukaryotic cells are called histones. And so in this picture, the DNA would be like the strand, the line would be DNA. And then the circles you see, each one of those circles would be an individual histone protein. And those histone proteins are able to attach to the DNA, but they can also detach from the DNA. It's not a permanent binding between the DNA and the histones. And those histones are going to group together into these groups of eight. And so this octamer, octa for eight, like octagon, means that we got eight histone proteins. But of those histone proteins, there's four types. So we got two each of four different types of histone proteins for a total of eight histone proteins that are going to come together in this octamer, and the DNA is wrapped around that octamer. And that bundle of one octamer plus the DNA is called a nucleosome. So a nucleosome is one little blob looking thing, and they're going to be back to back. You don't have just one of them. There's lots of those. Every 100 base pairs or so, you're going to see this group of a nucleosome. And so because we've got a lot of them, what they're going to look like is beads on a string. They're going to look like beads of proteins that are found on this string of DNA, kind of like this. And so we start out with the DNA. The DNA is bound to the histone proteins. The histone proteins wind around the DNA so that way we can make this nucleosome. But then we have multiple loop nucleosomes, and they're all more or less evenly spaced. So we got nucleosome, nucleosome, nucleosome as we go down the line, and we get these beads. And so those nucleosomes are going to zigzag back and forth from here to here to here to here. And that's helpful because it's going to create structure and strength, right? That means that the DNA is wrapped around the histone proteins, but then the histone proteins are interlocked with each other so that everything is nice and rigid and protected. And that's going to overall shorten the total length of DNA sevenfold. And when you get these zigzags like this, where the nucleosomes are stacked on top of each other like that, we call that a 30 nanometer fiber because the width from one side to the other is 30 nanometers. So we got the primary structure of DNA, those individual nucleotides. They're wrapped around histone proteins to make nucleosomes. The nucleosomes are then organized with each other to make these 30 nanometer fibers. The 30 nanometer fibers, so now this is not just DNA right here, right? This is DNA plus those histone proteins that made almost like a linear shape, right? So if we zoom out, imagine now you got this new string looking thing, that would be this. Those are then looped together, bundled with these proteins that are called SMC proteins and CF binding factors. And so they're going to help to take that long stretch and then loop it. Take that long stretch and loop it. The way I imagine this is, let's imagine you got a rope or an extension cord and you're going to wind it up. You probably are going to like grab it with one hand and then you pull it towards yourself and make a circle. And then you pull more and you make a circle and you make a circle, right? And as you're making a circle, your hand is grabbing that loop, that winding of rope, right? And so this protein right here would kind of be like your hand grabbing those loops. And that is going to happen over and over again. And that is how we're going to compact the DNA and also keep it organized. And so, again, the order that we're going to go in is DNA double helix. DNA double helix is round with histone proteins to make nucleosomes. The nucleosomes are then organized with each other to make these 30 nanometer fibers. And then the 30 nanometer fibers are organized into loop domains. Like imagine it's an extension cord that you're like holding it into these loop domains, which is about 300 nanometers. And then that altogether is going to lead up until our metaphase chromosomes. But remember that when we talk about metaphase chromosomes, those chromosomes are only going to exist while the cell is actively dividing. While the cell is living its life during the G1 phase of the cell cycle and doing most of its daily cell stuff, this would be the most common form of compaction. It would stop at these two levels of either euchromatin or heterochromatin in terms of how much compaction there's going to be. So let's look at the difference between those. So those words, heterochromatin and euchromatin, those have come up before. And if you're looking at the DNA, there's going to be areas where the DNA is more dense and areas where it is more loose. Remember, where it is more dense, we call that heterochromatin. And then where it's more loose, we call it euchromatin. The way I suggested you remember it, the way I remember it is heterochromatin, I think hard, like heterochromatin is dense, like it's hard. And so these would be the heterochromatin areas. And if they're really dense like that, it means that enzymes and proteins cannot access that DNA because it's wound up so tightly. It doesn't mean that it's like broken or something. It just means that it's like all your stuff's inside of a box. You can't actually use it because it's all packed up right now. You could always unpack the box. You could always relax these areas and stretch them out. But the fact that they're wound so tightly and so densely is letting you know that the genes, if there's any genes at all in that area, are not used very frequently. And that at that particular moment, they're just not needed. And so that's why it's a heterochromatin area. So heterochromatin in general, we say, is transcriptionally inactive. It's just not full of genes. But then the euchromatin areas, these are transcriptionally active areas. These are areas where you do produce genes. There are coding regions within euchromatin areas. So the okay. And so that is going to be the difference between heterochromatin and euchromatin is do you make these loop domains or not? Because that's what this figure is showing you over here, is that if you're making the loop domains, you're at the level of euchromatin. But then if those loop domains become compacted further than that into what we see right here, these 7 nanometer wide regions, now we're at heterochromatin. When we get to a metaphase chromosome, everything is temporarily heterochromatin. It's all super condensed because we're trying to move the entire chromosome around at that moment. But again, that's not going to exist for most of the cell cycle. It's only while the cell is actively dividing. When the cell is hanging out for most of its life during G1, it's going to look something more like this, and that the chromosomes are relaxed, the DNA is stretched out. They're not going to look like X's. When you stain them, they're going to look like these fuzzy blobs. And it just means that the DNA is opened up and we're using it to make new enzymes. And so they're still going to be separated more or less by the chromosomes. So in this picture, what they're trying to show you is that they have each different homologous pair stained a different color. And then when you look inside of the nucleus during interphase, when the cell is using those chromosomes to make stuff, even though they are not condensed, into that X-looking shape that we would see during metaphase, they are still at least separated. So like your chromosome number 1 information is going to be spread out, but it's not intertwined or bleeding into your chromosome 2 DNA. Because again, we don't want them to be so relaxed that they actually get tied up with one another and now we can't compact them anymore. And so there still is separation, and we call it their chromosome territory. And they're shaped more or less in a circle because this picture would have been taken within the nucleus. And so they're spread within the nucleus, and the nucleus has a circular shape. That membrane of the nucleus has a circular shape. And so this is how we're going to organize eukaryotic DNA because again, there's so much of it. In this picture, it kind of takes a little bit to understand what you're looking at, but this is a really beautiful picture. This is a picture of a chromosome that has been exposed to really, really concentrated salts. And when you do that, those charges within the salts, because remember, salts they're all ions. Those charges are going to help break apart the uh, bonds between the DNA and the protein to where now this would be those histone protein scaffold. This is like the proteins that used to be there to make up the chromosome shape. They're all hanging out, but all the DNA is disassociated from it. It's separated. And so all these squigglies you see in the background, this is all DNA. So this would be like an individual strand of DNA. So this cell is dead. I mean, to do this, you're going to kill a cell. But it's just a really cool way to show how without those organizational histone proteins that are creating this scaffold, all of the DNA would just kind of go everywhere and it'd just be a total mess. And so DNA is very, very highly compacted. It would not fit inside of the cell without this compaction. And the proteins that are involved with compacting that DNA are called histone proteins. And then we have this hierarchy of organization, which is histone proteins to nucleosomes, nucleosomes to a 30 nanometer fiber, 30 nanometer fiber, which then have loop domains. And then that loop domain structure can be condensed again if it's going to be something like heterochromatin or metaphase chromosomes.